Okay, so we're going to get started again. And yeah, and what I'm going to do is provide some background on biological macromolecules, and specifically on RNA, which should answer some questions that arose earlier. And then Matt is going to talk about RNA folding and how we can predict what this interesting molecule is going to do. So what is it? What does it do, and why do we care? Way back in 1958, Francis Crick proposed the central dogma of molecular biology. He later admitted that he really didn't understand what that word meant, but it kind of stuck. And basically what it's doing is describing the flow of genetic information, where DNA is the repository and DNA is copied into RNA, and then the RNA is used to make protein. And that's a reasonable first pass at this question. But it presents really only a fraction of what RNA is doing. Yes, absolutely, RNA is involved in this process as an information carrying material. It's involved in the translation process. It makes up the machinery, the ribosomes that are, of course, proteins too, that are involved in protein synthesis. RNAs are involved in processing of the initial RNA transcripts. They're involved in control of gene expression in a variety of ways. And basically, the more we learn about cell biology, the more we learn about roles, new roles of RNA that we didn't know about before. So the central dogma was just the beginning. Just a few words about what these molecules are. DNA and RNA are both nucleic acids. They have pretty similar structures. They're polymers of nucleotides. DNA is, I'm sure, better known, right? That's the genetic material in all cells. RNA facilitates protein synthesis and has roles in the control of gene expression. And it's similar to DNA in some important ways that we'll talk about, but it's also different in important ways. And then there are other proteins. Proteins are workhorses of the cell. They're responsible, well, the enzymes that we've spoken about previously, like beta-galactosidase, that's an enzyme. The transport protein, the lactose permease, that's a protein. Proteins are involved in control of gene expression also, and they're responsible for cell structure and motion and all kinds of wonderful things. But they're not the only enzymes. There are RNA enzymes called ribozymes. So the more we learn about RNA, the more interesting it becomes. Nucleic acids, they're just polymers of nucleotides. And a nucleotide is a nitrogenous base, we'll meet them in a minute, a sugar, ribose or deoxyribose, and phosphate. Okay? DNA has deoxyribose. Again, I'll show you a picture in a minute. And its nitrogenous bases are A, C, G, and T. And you all knew that, right? You've seen a science fiction movie? Yeah, absolutely. RNA has ribose instead of deoxyribose, and it has A, C, G, and U instead of T, like we talked about this morning. DNA is double-stranded, and the association is due to hydrogen bonding between those nitrogenous bases, and RNA is single-stranded, and that's going to be important. So here are the pieces, A and G, and C and T and U. Here are the two sugars. Ribose has a hydroxyl group here. Deoxyribose doesn't. And then phosphate, there's one. Can be strung together depending on what we're talking about. So how do they go together? There are two kinds of bonding that are important here. One will be the covalent bonding that holds the polymer together. Right? So that makes up the backbone, and that's the sugar phosphate backbone. And the sugar phosphate is what that P in CPG represents. In this case, it's DNA, so it's a deoxyribose and a phosphate, and then there'll be a C with a deoxyribose phosphate, and that'll be attached to another that has a G. The nitrogenous bases are kind of sticking out from that backbone. They can form hydrogen bonds with other bases. If we're talking about DNA, that's pretty much what they do. If we're talking about RNA, that's what Matt will be talking about. The strands are directional. One end is not like the other. One end has a phosphate, the other end has a hydroxyl group. And by convention, when we write a sequence, we write from 5' to 3'. So if you see this, it means that. Okay.
Sorry.
Yes.
That last one, 5' to 3'.
Okay. It's a directional molecule, right? So let me show you what that means. 5' is where the phosphate is up there. 3' is where a hydroxyl group would be here. But what does that mean, right? Why prime, right? Well, when we number them, we number these, 1, 2, 3, 4, 5, et cetera. And to distinguish whether we're talking about these or these, we call these 1', 2', 3', 4', 5'. Okay? So when you say 5' or 3', you're talking about something that's on the sugar. Okay? And the way the molecule goes together, there'll always be an end that has a 5' phosphate, and then there'll always be an end that has a 3' hydroxyl. Okay? Does that make sense?
So the 3' is the position? Yes.
Yeah. So 1', 2', 3'. 3' hydroxyl. Yeah. And then on that one, let's see, where's my pointer? Right. 1', 2'. Ooh, that's missing its hydrogen. 1', 2', 3', 4', 5'. Here's the phosphate. Pardon? Oh, whoops. Sorry. Where did we go? Yeah, each vertex is a carbon, right? So that's a carbon, that's a carbon, that's a carbon, that's a carbon. Here we actually specify that carbon. But it's 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbons. Ribose and deoxyribose are 5 carbon sugars. And to keep these separate from those, 1', 2', 3', 4', 5'. Okay? All righty. So that's the general structure of one of these polymers. What kind is this? Yeah, that's DNA. How do we know? Because it's got a T, and there's no hydroxyl group there. Okay? All right. So here's the way you're used to seeing DNA. The double helix, they're anti-parallel. So this one goes from 5' down to 3', and that one goes from 5' up to 3'. And then here are the hydrogen bonding patterns between the bases. So G always binds to C, and A always binds to T. Okay, pretty straightforward. There it is. Over here is RNA. You'll notice we have hydroxyl groups here, and we have a U instead of a T. The other thing that kind of jumps out at you is that this is single-stranded, right? And that means that this has a lot more freedom in terms of the three-dimensional shapes it can take up. Okay, and that's going to be, well, that's why this is so interesting, right? DNA, if it's hydrogen bound, it makes a helix. You can have it a little tighter or a little looser, but it's still a helix. RNA can do all kinds of interesting things, some of which are functional. Okay, I stole this slide from Matt because it has a mathematician's definition of RNA. right? See, right up there. Combinatorially, an RNA strand is a length end sequence of bases or nucleotides over the alphabet A, C, G, and U. I would never have said that. That's not me. That's a mathematician's definition.
Before that, I had all the biological slides that were like undergraduate versions of what you had before. This is like my slide 8.
Yeah, but I mean, it's great, right? It points out the different way that we think about these things.
I tried to do
Okay.
the biological one first, but it probably didn't come off too well.
No. See, I mean, here, this is your slide.
Let me ask, does
Yeah.
that definition indicate directionality as well?
Yes,
Does it?
It's a
Oh.
sequence, so 1, 2, 3.
Oh, okay. See, I learned something. Yep, I learn every time I do this. Okay, good. So the other thing that this provides us with is the bonding that can happen in RNA. So since there's no T, RA will bind with a U. G and C do the same thing that they do in DNA. And then there are these funny things that can happen. So here's a wobble pair with G and U. This is the most frequent funny thing that can happen, but there are some other oddities that RNA can do. RNA is a nonconformist. Okay, so how does RNA fold? These are all actually the same RNA sequence. This was put together by Christine Heitch. All the same, but these are all the different things that theoretically that particular RNA can do. Ooh, yeah, very interesting. Don't know what they mean, but they're very interesting. Okay, but here's one I do know what it means, and this is also from Christine Heitch. So here is a sequence of a real RNA. So this is the primary sequence, and we start here. That's a 5' G, and we read all the way down to 3' down here. Okay. This is a representation of the secondary structure. So these are the hydrogen bonding patterns that we can find in this molecule. And they're laid out flat. This is called a cloverleaf model. But you can see G's binding to C's, and here's a GU, and there's an AU, and so on. Okay? This is what the molecule really looks like. It kind of folds over onto itself, and then it plugs into a little slot in the ribosome, bringing with it an amino acid in the process of protein synthesis. This is a transfer RNA, and that's the functional structure. Right? This is the one that does something useful for the cell.
So you leave that slide. What are the 10s, 20s, 30s?
Oh, that's just keeping track of 1, 2, 3, 4. Yeah. So here's our 5' G, right? And then you just walk your way around. Okay. This is the end where the amino acid gets attached. That CCA. Yes. Yes. Yeah. So this, it's actually upside down. This is this little bit down here, right? And then this is that right there. Okay? Yeah, it's there. But yeah, you kind of take and go. Okay. Next one. Alrighty. So sequence, secondary associations due to hydrogen bonding, and then the final functional three-dimensional structure. I'm going to show you one more model like this. Okay. And again, here's another cloverleaf kind of a model. Here's the nicely folded model. Again, here's the business end. Here's where it binds to the messenger RNA. And then this just gives you more of an idea of what that thing really looks like. Isn't it cute? Yeah.
So, can you explain what you mean by modeling? Because when you say model means something else to me, something you can create. So, are these things being done by whoever was doing this?
It's like model train.
Yeah. This is not a model like you think of a model. This is like something I would make with clay. Okay? Yeah.
Where is that in the right-hand picture? Where is that?
Okay, so this is this loop here,
Okay.
right? Because that's where this molecule is going to interact with the messenger RNA that has the gene sequence in it.
Like stuck inside or something?
Oh, no. What? Okay, so here, and then there's that little curve here, and that little curve, and the little bit sticking out here is where the amino acid attaches. Yeah.
What is that cylinder thing?
That cylinder is representing this. That's all. Yeah. And biologists tend to do this. You know, when we talk about models, we mean like physical models. I make them with clay and, styrofoam and you name it. Whatever I can do to get my students to build a mental model, I will physically construct these things. And the reason for that is that all of these associations are based on shape, right? Structure determines function, whether we're talking about proteins binding to other proteins or enzymes binding to their substrate. It's very much a physical association. So yeah, and this is one of those words that we use very differently. Yeah. Okay. So here we go. Yeah, why? What's important here? Why do we need help with these interesting molecules? Well, there are two really important questions about RNA folding. Question number one, given an RNA strand, can we predict how it's going to fold? That's an important question because that folding pattern is going to determine its structure, right? And that's the second question. What's the relationship between the structure of this folded RNA molecule and what it does in the cell, okay? So this one is the biological question, but since we are learning more and more about these RNA molecules, it would be really helpful if we had some clue about question number one. But question number one is where the mathematicians and the computer scientists and the quantitatively skilled people can help. Okay? And this is my cue to turn. Yes, I turn it over to a quantitatively skilled person.